Welcome back, fellow mitochondriacs, for another episode of Cancer is a Mitochondrial Metabolic Disease. We're going to continue our discussion into enzymes and proteins that are responsible for converting glucose from its original form down to its metabolized fermented forms through the glycolysis system, or the aerobic glycolysis system, called the Warburg effect. We spent the last couple of videos looking at how to manipulate the ability for glucose to actually enter the cell through these GLUT transporters. And now we're going to look at the first step in glycolysis and an enzyme called hexokinase. So hexokinase, I think it's interesting because a lot of times medicine or conventional research will look at one very particular target, and they will look at things to inhibit it. They'll look at its fundamental value for a particular disease process. And I think that it's it's very interesting to look at one particular thing. What we're looking at is down here. We just left these little micro series about the GLUT transporters, which is how glucose actually gets into the cell. But we're now talking about hexokinase. And again, it's interesting because these researchers will look at one particular thing. Now, some of these papers will look at glycolysis as a whole, but my point is that we're going to zoom in specifically to hexokinase today. But we want to ultimately shut this entire process down, right? We talked about this in the past. We've looked at utilizing the ketogenic diet as a foundation for metabolic therapy. We've looked at ways to lower blood glucose by allowing it to be kind of absorbed by other tissues that are not cancer cells. And then we've most recently looked at ways at potentially inhibiting either the expression and placement of these onto the cell membrane itself, or things that actually block this transporter here. And we're going to go to the next thing. But obviously, when we zoom out and we say, hey, like we want to shut this process down essentially completely. We want to be fat and keto adapted so that the other cells are minimally or not at all injured whatsoever. We know we're going to be looking at multiple targets for our therapeutic approach. But it's just interesting because you're going to see research kind of like talk about how important even just one little piece of this is for the goal of slowing down or killing cancer cells. Like, for example, like I was able to present a lot of information just about just one transporters. And you're going to see the same thing about hexokinase, the same thing about PKM2, lactate dehydrogenase, PDK, as well as hypoxia factors. And it's going to make it seem like that one particular target is the be-all, end-all. And I'm not saying it's not important by all means, but my point by kind of giving you this gestalt message is that we're going to be trying to have multiple targets at once. So that way there's redundancy. That way, if there's any escape around that particular pathway, then we can hit it further down. So that way there is a higher chance of success. Anyway, again, we're talking about hexokinase today. So the first paper I pulled was from 2015 in the journal Autophagy, and it says hexokinase 2 integrates glycolysis and autophagy to confirm cellular protection. And it says hexokinases catalyze the first step of glucose metabolism, phosphorylating glucose to glucose 6-phosphate. Hexokinase 2 is a predominant isoform in the insulin-sensitive tissues such as heart, skeletal muscle, and adipose tissues, and it is also upregulated in many types of tumors associated with enhanced aerobic glycolysis, aka the Warburg effect. Accumulating evidence indicates that hexokinase 2 plays an important role not only in glycolysis, but in cell survival. Although there is increasing recognition that cell metabolism and cell survival are closely related, the molecular link between metabolism and autophagic pathways has not been fully elucidated. We recently discovered that the HK2 facilitates autophagy in response to glucose deprivation, hexokinase substrate deprivation. Basically, what they're saying is that when you restrict glucose or deprive the cells of glucose, then hexokinase, its only role is to take glucose and turn it into glucose 6-phosphate. You're depriving its substrate to protect cardiomyocytes, and suggests that HK2 functions as a molecular switch from glycolysis to autophagy to ensure cellular energy homeostasis under starvation conditions, which I think is pretty interesting. So what it's saying here is that when we're in a nutrient-rich environment, then mTOR will be activated through the PIK3-AKT system via amino acids and insulin and different things, and it's going to produce energy. And this hexokinase is going to be converting that energy into the next steps of metabolism. However, when you deplete energy and there's a depletion of mTOR and AMPK is activated, then that is what is going to be at least partially upregulating autophagy for these cells. So this group was able to find out the link there back in 2015, or suggest a link. Later on, I pulled this paper because it's so good, even though it's quite a bit later than the next paper. It's published in December of 2023 in Molecules, and it said the promoting role of hexokinase 2 in tumor development and the research progress of its inhibitors. And it says here, increased glycolysis, or the breakdown of sugar, is a key characteristic of malignant cells or cancer cells that contribute to their high proliferation rates and ability to develop drug resistance. The glycolysis rate limiting enzyme, hexokinase 2, is overexpressed in most tumor cells and significantly affects tumor development. This paper examines the structure of HK2 and the specific biologic factors that influence its role in tumor development, as well as the potential HK2 inhibitors in anti-tumor therapy. And what we see here is kind of an inversion to some degree of this here. We see that glucose is being brought in, glucose is being converted to glucose 6-phosphate, or G6P, from hexokinase, the enzyme. It converts that. And what you're seeing here is how glucose starts here, hexokinase 2 here is being converted to G6P here, right? It's just an inversion of that what we've already seen. Well, I pulled this particular paper because it had beautiful graphics and it just really had a great explanation of kind of the importance of hexokinase. And I particularly thought this was interesting because it's showing all of the different factors that are influencing the hexokinase enzyme expression. And that is going to be seen as an RNA, right? And then RNA is going to get transcribed to a protein, or translated to a protein, excuse me. And that will make the actual functional enzyme, but that functional enzyme will have to be transported to the cell, to the cytoplasm so it can do its job. And it's under the influence of multiple pathways, the MAPK pathway, beta-catenin. We have STAT3, things that I've heard of before, the PIK3-AKT mTOR pathway. NRF2, interestingly enough, is influencing this. The androgen receptor kind of shows how, sex steroids, such as androgens, and some particular cancers, or even estrogen receptors, probably also are responsible for this as well. And then there's, of course, something that we've talked about a lot, hypoxia-inducible factors. And hypoxia-inducible factors are stabilized not only by hypoxia, which is what's classically how it got its name, but by several other factors, including inflammatory cytokines such as IL-1 beta, as well as many other factors. That's called pseudohypoxia. So when you have a lot of inflammation around, that can upregulate and stabilize hypoxia factors, which can lead to upregulation of not just hexokinase. We've seen graphs where the hypoxia factors, as well as some other pathways, can upregulate all of the necessary kind of machinery, with hexokinase being one of those machineries for the Warburg effect and for the enhanced glycolysis, or the enhanced glutaminolysis that's seen in the cancer phenotype of increasing glucose and glutamine metabolism for the fueling and growing of cancer. But this is just showing how many things are influencing hexokinase, just that one particular enzyme. But it's not just any enzyme. It's a very important rate-limiting enzyme within the glycolysis system. The paper then shows, again, basically it shows this in a non kind of graphical representation, where you have multiple pathways, including MYC, which is not on that first graphic. Yeah, it's pretty unbelievable what can upregulate this thing, and that's a lot of times why, you know, medicine gets into wanting to block one particular pathway, right? Maybe they want to block the STAT3 or the c-MYC or the cyclin D or the androgen receptor as a way to kind of upstream affect this, when there could be multiple things in place. And I think that it's still going to be kind of a losing strategy, because once you start to affect one particular pathway, then particularly another pathway can take its place. And we see that it's influenced by many things here. It's also downregulated by many things as well. Not quite as many things as it being upregulated. And most of these are like microRNAs. So there is an association between hexokinase and tumor drug resistance. And it says here that hexokinase expression is upregulated in tumor cells, granting them metabolic advantages and inhibiting apoptosis. This leads to enhanced cell growth, increasing their resilience against chemotherapy. It says, moreover, that trastuzumab-resistant gastric cancer cells, hexokinase 2 levels were found to be elevated to display a diurnal oscillation pattern. I think it's interesting that there is almost a circadian rhythm to this, a diurnal pattern. However, the viability of trastuzumab-resistant cells can be suppressed through the inhibition of hexokinase 2 using substrates such as 2DG or metformin, ultimately enhancing cell death. As a result, the combination of hexokinase inhibitors with chemotherapeutic agents holds great potential as a robust approach for effectively treating tumors. So what they're saying is that the higher that these metabolic enzymes, such as hexokinase, are upregulated, the more likely there is to be chemo resistance. Well, that's a big problem, because most of these cancers are utilizing the Warburg metabolism. That's why it's a hallmark of cancer. And in order for the Warburg effect to functionally work and to be able to be metabolizing 10 to 30x times more glucose than you would under normal conditions, you're going to need a lot more enzyme to metabolize all that substrate. So therefore, it's likely going to be the case that many of these cancers are going to be chemo resistant. And so what it's saying here is that, and we're going to hit on these, okay? So don't need to worry about the actual particular substances. We're going to have a whole thing today on metformin, and then we'll talk about 2DG probably later this week, because there's such a big topic. But they were saying that when you use things like 2DG or metformin, then that enhances cell death. And then when you use those kind of things in combination with chemotherapeutic agents, that's why I have personally not seen any literature to actually suggest that if you were to do metabolic therapy, that would inhibit the standard of care whatsoever. As a matter of fact, I've only seen that it has either minimal effect at enhancing it or a significant effect at enhancing it. Or even, moreover, I've seen it where it will take a cancer cell that is basically resistant to either radiotherapy or immunotherapy or chemotherapy, and it'll actually unlock that resistance and make that particular tumor sensitive again. So there's really no downside to looking at cancer as a metabolic disease and starting to attack it in such a way. So there's a drug synergism between kind of suggested in that last one, the last little snippet. It said, unfortunately, the inhibition of specific cellular pathways is unable. I put this in because I think it highlights that we can't be looking at this in one particular, like we're going to hit hexokinase today, and it's important. I don't want to minimize that, but to just be looking at blocking hexokinase in a vacuum is not going to be likely a winning strategy. It says here, unfortunately, the inhibition of specific cellular metabolic pathways is unable to completely impede the proliferation of tumor cells. The presence of metabolic reprogramming in cancer allows them to adopt alternative pathways for cellular metabolism, ensuring survival. Consequently, a more efficacious therapeutic strategy lies in the adoption of a multi-target approach that utilizes drug combinations instead of solely targeting the glycolytic pathway. I would say that I don't know if I necessarily agree with everything they're saying in that particular snippet. I think that the wisdom is don't focus on one particular enzyme and think you're good, right? I think that if you're looking at really shutting this process down and knowing that the Warburg effect and glycolysis, aerobic glycolysis, is the Achilles heel or one of the Achilles heels of cancer, in addition to the enhanced glutaminolysis or the glutamine utilization, then it makes sense to attack glycolysis and sugar utilization at multiple steps. That would be first and foremost, again, a ketogenic diet, things that lower blood glucose, things that steal glucose away from cancer cells, things that block that first step of bringing glucose into the cell, and then later having a multiple target approach of glycolysis as well. Now, I'm not saying that would even be enough by itself. That would probably be a much more effective way of just then looking at just hexokinase or just GLUT transporters, right? But still, you got to take care of the glutamine side of things as well. And it would make sense in my view, at least, that you would have a multiple target approach for the glutamine as well. So being on a therapeutic ketogenic diet, you're not on a very high protein diet in general. So there's not going to be a lot of amino acid, being brought in from the outside, from the eating and from the dietary intake. But you can't stop it that way. We've talked about that in the past. Glutamine can be made by the body, right? So then there's other ways like exercise and other things that can actually steal kind of glucose or glutamine out of the system, out of the bloodstream. And then there's particular enzymes or transporters that bring it in. There's enzymes that utilize it, whether it be glutaminase, whether it be glutamine dehydrogenase, et cetera. And so my point is that there's wisdom in what this author or these authors are saying. It's just that what their view is that don't just look at the metabolism of cancer and treating it as a metabolic disease. You need to also be looking at the genetic pieces and using chemotherapy. And of course, that's their perspective. I think that in some ways does make sense. But if there's a way to attack the Achilles heels that the other cells don't have, then it makes more sense for me to utilize those Achilles heels instead of using things that chemotherapeutics can affect the normal cells as well. Radiation therapy can affect the normal cells as well. So you're having a much less therapeutic window, and you're not really attacking the Achilles heels, in my opinion. With that, I'll digress. I'll go down further here. So other anti-tumor drugs, and I particularly highlighted here that studies have also shown or found that metformin acts as an inhibitor of hexokinase 2, further supporting its significance in cancer treatment. So not only does metformin have effects at lowering blood glucose, enhancing AMPK, and particularly shutting down at least one of the pseudohypoxic states within the body. It also we've also looked at it blocking part of the glutaminolysis pathway with the SLC7A11 particular glucose cysteine antiporter, and helping to kind of diminish the glutathione that's being made within cancer cells. Well, now we're seeing that it has another role at inhibiting hexokinase 2 as well. So it says here that targeting glycolysis to enhance immunotherapy says arguably immunotherapy is now being considered one of the most promising therapeutic strategies for several types of cancers. And immune checkpoint blockade, ICB, and immune adoptive cell therapy, ACT, based therapeutic strategies have been approved for several cancers. However, several reports have shown that a high percentage of patients initially fail to respond to these interventions or acquire resistance in the long run, limiting the application of these promising strategies. Several studies have reported that metabolic reprogramming cancer cell that leads to development of immunosuppressive tumor microenvironment. It is one of the main contributors to the reduced efficacy of immunotherapy. Further, it has been suggested that metabolic interventions can significantly enhance the efficacy of immunotherapy. So this is just not me, but others parroting, or not parroting, that's not a fair way to say it, but I'm parroting what they're saying, right? What I'm trying to say is that when we are affecting glycolysis or glutaminolysis, we are enhancing the traditional therapies, okay? And even these like newer therapies, like these checkpoint inhibitors and these immunotherapies, they have, what is that here? They've shown to have a high percentage of patients who initially fail them because they acquire resistance. And what is leading to the resistance? It's the immunosuppressive tumor microenvironment, the same problem that affects chemotherapy, the same problem that affects our own immune system, right? It's the acidic tumor microenvironment that shuts down success. So therefore, when we shut down glucose availability on a ketogenic diet or by utilizing other strategies that block glucose from being available in general, we're shutting down lactic acid production. When we block GLUT transporters, we're shutting down lactic acid production. When we are blocking hexokinase or lactate dehydrogenase or other pieces of the pathway, we are shutting down lactic acid production. When that lactic acid production falls off a cliff, the tumor microenvironment now is not as acidic as it was. Now these things can work. Now our own immune system can work. So therefore, this is a big part of the strategy of how metabolic therapy actually works. It's not just about shutting down ATP. It's not just about shutting down glutathione production and allowing these cells to be susceptible to oxidative therapies. It's also the fact that these strategies of metabolic therapy turn on the immune system. They allow the immune system to actually function like they're supposed to. That way they can get to where they need to go and do what they need to do. And there's another piece here that I highlighted from the paper, targeting glycolysis to improve immunotherapy. And it says the interplay between anti-tumor immunity and cancer metabolism suggests that combining immunotherapy with glycolysis-based therapy is a promising strategy to improve treatment efficacy. Let's be honest, it's a promising strategy to improve your success with cancer in general. That's why metabolic therapy is the foundation. It should be the foundation of all cancer therapy. Things like chemotherapy, immunotherapy, radiation therapy, in my opinion, should be way down the line once you have maximized metabolic therapy, and that will probably be, God willing, in the future, what all is needed, to be honest with you. And these other therapies will probably be not needed unless you are in dire straits in like a salvage type situation, or maybe for like induction to get yourself like back on track if you're really in bad shape when you start or something like that. But I think that it's becoming more and more clear as I see more and more research that metabolic therapy has to be the foundation, and the industry is probably going to not be on board with this because it's so effective. But medicine, doctors who care about their patients need to be up on this. They need to start to implement these, know how to implement this, how to track this. It's not rocket science. You don't need to be, you don't need to be a biochemist. Any medical doctor could implement this safely with just doing a little bit of refreshing on the basic biochemistry that they get in medical school and undergrad. Later, it says, it has also been shown that blocking glutamine metabolism increases T cell function. Furthermore, supplementing the culture medium with glutamine the glutamine antagonist, DON, 6-diazo 5-oxo L-norleucine, enhanced CART cell fatty acid oxidation and reduce glycolysis, making the CART cells remain in a more undifferentiated state, inhibiting glycolysis using hexokinase 2 inhibitor 2DG before ACT, that's an immunotherapy, induced a memory-like phenotype in the T cells, enabling more efficient control of tumors and prolonged survival in animal models. So I put this very interesting tidbit. I pulled this because I thought this was, it's giving a little bit more of a piece of this to the story, okay, that I think is necessary within cancer cells, okay? So, or within cancer kind of circles. So one of the big fears is that when we restrict glutamine from the immune system, we're going to shut the immune system down and it's going to be ineffective at doing what it needs to do. And I think the fear is valid. But the issue is that there's been papers that I've seen myself, and then it's kind of highlighted here. When you restrict glutamine, it actually enhances T cell function and other immune cell function. And it says here that when they gave DAWN to these T cells, it actually enhanced their reliance on fatty acids, and it reduced their need for glycolysis, which I think is also interesting because how would blocking glutamine reduce glycolysis? I'm just spitballing here. Maybe I've seen where glutamine, when it gets broken down to ammonia, and I want to say it's glutamate or alpha ketoglutarate, that ammonia can stabilize hypoxia factors, and that hypoxia factor stabilization may be able to enhance glycolysis. I'm not really sure what the interplay is there. I think that I need to look that up. But I think this was very fascinating. When they gave DAWN, the amount of glycolysis or glucose consumption dropped, and fatty acid oxidation improved within these T cells, just the immune cells. Then what they did was they inhibited glycolysis using 2DG, 2-deoxyglucose, which we're going to have a whole video on. And it induced a memory like a memory-like phenotype, or a type of T cell that had a stronger memory. And it enabled these T cells, when you block the glucose via hexokinase blockade via 2DG, it made them more efficient at controlling tumors and at prolonged survival within animal models. So there's more to this story, especially when it comes to the metabolic therapies effect on the immune system, that I don't personally know, that I would love to learn, but it's starting to become more clear that it seems that everything works better under therapeutic ketosis, is what it seems like to me, including our immune system. Furthermore, a transient glucose restriction followed by a glucose re-exposure could enhance the tumor clearing efficacy of CD8 cells. So I particularly highlighted that because it makes me think that when we have enough maybe data, or when we have enough tools in our tool bag to be using to effectively shut down glycolysis and glutaminolysis, maybe, just maybe, the ketogenic diet, or at least maybe not the ketogenic diet itself, but maybe the ketogenic diet in extremes where we're having glucose ketone indexes of like 1 or less, blood glucoses of 70 or 80 or less. This makes me think that maybe the severity or the strictness of the ketogenic diet could potentially be cycled. In fitness community and in the bodybuilding community, they, in order to lose more fat, a lot of times they'll do carb cycling, and maybe carb cycling could be a way to enhance anti-tumor immunity. And the reason I say this is because it says once you restrict glucose and it was followed by re-exposure of glucose, basically you take it away and you give it, that could enhance tumor clearing efficacy of some of these T cells. So Could glucose, in addition to glutamine, be pulsed as well? Maybe. And maybe that could be a good thing. Time will tell. I think it may be difficult, though, that being said, just functionally and clinically, to be on a strict ketogenic diet and then let up, and then strict ketogenic diet and let up, and then strict ketogenic diet and let up. That may be difficult. Maybe we're talking about not so much the diet itself. Maybe we're talking more about glucose inhibitors. And that's why, if you look back, gosh, I don't even know if I have the slide, not readily available, but if you look at the initial press pulse slide from Seafried, he does pulse glucose inhibitors. It could be for this very reason. Maybe there's more method to his madness that I'm unaware of. All right, so I wanted to, in particular, also highlight this. Again, this is why I pulled this paper. It's so good. Targeting glycolysis to enhance hormonotherapy. So I think that breast cancer and uterine cancer and things like that, even ovarian cancer, there are a lot of publications that allow us to, I guess, maybe more easily believe that metabolic therapy will be helpful for those cancers. But it seems that there is a particular hormone-driven cancer, prostate cancer, that is a little bit maybe more cautious or on edge when it comes to being on a ketogenic diet for that particular type of cancer, because there are publications that suggest that fatty acids can be used by prostate cancer cells. And, I've had dozens of people send me these publications. And when you actually look at the publications, the ones that aren't, behind a paywall, and you look at what they're trying to say, from what I can tell, it seems that most of them are saying that cancer cells can use fatty acids, not as fuel, as we would think of like being used in a mitochondria to make energy through the electron transport chain, but more that the fatty acids are being used to make other fatty acids to proliferate, which is true. All cancers do that, but not necessarily in the way that it's being kind of like, I don't want to say fear-mongered, but kind of fear-mongered that prostate cancer is like a special cancer that has intact mitochondria that can burn ketones, that can burn fatty acids for fuel. That I have not seen be true. And it kind of goes against everything we know about cancer in general, like that, mitochondria are damaged, and mitochondria that are damaged, structurally are not going to function. We know that, looking at ATP studies and et cetera, it just doesn't make sense. So I do believe there is a continuum. We've talked about that at length in the past, how there's a continuum of let's say this is at the top left here for me, the reliance on the mitochondria and having the metabolic flexibility and being able to use energy through OXPHOS is at the highest level in a normal cell. And as time goes on, there is a lowering of that because as the mitochondria get broken essentially, and then there is a low amount or no amount of aerobic glycolysis being used, and then over time that will go up, right? Well, there's going to be a point in time where both are being used, right? It's just that if you look at Seafried's work, they don't believe that even in these partially oxidative cancer cells, where there is some amount of oxidative phosphorylation going on, it's not enough to maintain the cells, right? So even if they're able to burn some amount of ketones or fatty acids or proteins or amino acids, right, through various, glucogenic and ketogenic pathways through the mitochondria, it's not enough. The defects are enough to where the cells can't survive. So when you're on a ketogenic diet, they can't, no cancer cells basically can survive on a ketogenic diet if you're blocking glutamine as well. So my point is that prostate cancer kind of gets a bad rap, and that's why I like to present these kind of papers that kind of like highlight that no, these cancer cells are really no different than the rest of the cancer cells, and it helps maybe give a little bit of reassurance. So I highlighted here, resistance to hormonal therapy invariably occurs in most patients with ER positive metastatic breast cancer and castration-resistant prostate cancer. Metabolic reprogramming is an inherent feature of endocrine-resistant cancer cells. An inherent feature, just like every other cancer. It's a hallmark of cancer, implicating that combination therapy with metabolic regulators and conventional hormonal therapy might be beneficial to overcome resistance. However, it is unclear whether metabolic rewiring is a cause or consequence of endocrine resistance. And several studies are investigating the crosstalk between hormone signaling and cancer cell metabolism. Elevated glucose levels resembling hyperglycemia in breast cancer cells has been attributed to the reduced response to tamoxifen therapy and could act as a marker for responses in hormonal therapy. AKA, if you're on tamoxifen therapy or estrogen deprivation therapy and you have hyperglycemia, that's bad. That's going to lead to resistance, and it's going to lead to failure of that therapy, which means that even if you're going to do conventional therapy, which is cool, do conventional therapy because that's what you feel is necessary for you. No one's going to, I'm certainly not going to tell you not to do any therapies or to do any therapies. But what I would encourage is, and highly recommend, is doing metabolic therapy in addition to that, because if you're not, then your conventional therapy is probably not going to have the long-term responses that you're going to be wanting, because ultimately most patients with these PR positive cancers or prostate cancers end up with basically a resistant tumor to those hormone blocking kind of drugs. An increased glycolytic rate is a characteristic feature of tamoxifen resistant cells, and inhibiting glycolysis is expected to restore the previous resistance to tamoxifen sensitivity. Elevated glycolysis in breast cancer cells is also associated with mitochondrial malfunction or dysfunction and upregulated AKT, mTOR, and HIF1 signaling pathways. This is not unique to breast cancer. Okay, this is an all cancer problem. Okay, it's talking about it in particular with breast cancer, and particularly, tamoxifen-resistant breast cancer. But this is all cancer. Tamoxifen-resistant breast cancer cells escape cell death by increasing autophagy. That's why autophagy is not all good. Autophagy is hijacked by cancer cells to escape cell death, just like glutathione is not all good. It's actually hijacked by cancer cells through the inactivation of the TOR via S6K via the hexokinase 2 pathway. Glycolytic inhibition by the knockout of HK or 3-BRPA treatment downregulated AKT mTOR signaling and could be a therapeutic strategy to overcome tamoxifen resistance in breast cancer. A recent study investigated the potency of a combination therapy employing low-dose tamoxifen, an estrogen receptor alpha antagonist, and metabolic inhibitors 2DG and CB839. How about that? That's basically like C-free in a bottle, right? 2DG and CB839 is improving the anti-proliferative effect of tamoxifen-resistant ERA-positive breast cancer cells. The triple combination showed superior cell growth inhibition by improving or inducing apoptosis or programmed cell death and c-MYC downregulation. However, the combination of tamoxifen with 2DG did not show any significant strong inhibition of cell viability. So it just goes to show that if you shut down glycolysis, glutaminolysis is still there, and therefore it's not going to have the effect you're looking for. It's going to be better than nothing, but it's not going to have the effect you're looking for. The triple combination of a glucose inhibitor, 2DG, with a glutamine inhibitor, CB839, plus tamoxifen is what showed the best effects. Now, I would argue that the best effects would be shown by probably an oxidative therapy such as IV vitamin C or HBOT or something like that, or maybe photodynamic therapy. But I'll digress. It's not talking about this in this paper. Later it says, in long-term estrogen deprivation model, the AI resistance, cancer cells were demonstrated to have increased glycolysis dependency. The inhibition of glycolysis with HK2 inhibitors, along with AI, such as and letrozole. So that's aromatase inhibition. That's what it stands for. AI stands for aromatase inhibitors, or aromatase inhibition, and letrozole is an aromatase inhibitor. Reduce cell viability. Dietary interventions that target metabolic rewiring has also been shown to affect endocrine therapy in metastatic breast cancer. Basically, metastatic breast cancer patients with liver metastases. Metastatic burden of the liver increases with increasing carbohydrate percentage in the diet. Ding, 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 right? Like the more carbs you put in the diet, the more metastases you have in the liver. Clearly, we're feeding the cancer with carbohydrates. The metastatic burden of the liver increases with increasing carbohydrate percentage of the diet. A fasting mimicking diet increased the efficacy of fulvestrant, which is another AI treatment, and it reduced the metastatic burden in the liver of these metastatic breast cancer patients, providing a proof of concept for a more straightforward strategy to circumvent drug resistance with a potential application in other cancer types as well, of course. So I think this is fascinating. So basically, fasting mimicking diets or carbohydrate restrictions, ketogenic diets, enhanced by itself the use of estrogen blocking drugs. In this case, are using either letrozole or fulvestrant, which are aromatase inhibitors. In the past portion of this study, they were looking at tamoxifen, which is actually an just to be very clear, it's an estrogen receptor alpha antagonist, right? So they both block estrogen, but at different ways, right? And when you took an estrogen blocker with just 2-DG, okay, in a vacuum, just blocking the hexokinase enzyme, you got really not much of an effect. When you took that glycolytic enzyme inhibitor, 2-DG, and you added CB839, which is essentially a glutaminase inhibitor similar to DON, and you added it to tamoxifen, there was an effect, a big effect. However, when they were using letrozole, they didn't use an inhibitor of just one part of the story, right? They didn't just look at hexokinase inhibition. They looked at kind of like just restricting glucose in general, right? Using a fasting mimicking diet or a low carbohydrate diet with letrozole, and it enhanced the well, first of all, it lowered the metastatic burden in the liver, okay, and it unlocked resistance to the aromatase inhibitors. So it just goes to show me, at least, that likely the ketogenic diet is going to be much more powerful, okay, than any one pieces of steps that are blocked by a specific inhibitor, right? So if I block hexokinase using 2-DG, that's not as powerful. Even if it's like complete blockade of hexokinase, it's not as powerful as being on a ketogenic diet or being on a fast mimicking diet, likely. Now, is there going to be probably an enhancement of the ketogenic diet if you add things like 2-DG or other hexokinase inhibitors? My thought is that yes, it's likely going to have an enhancement of that ketogenic diet, but the ketogenic diet really needs to be the foundation. It later says here in this paper, and I don't highlight it because I want to read the entire thing, that endocrine resistance remains a major clinical barrier that requires the development of novel strategies to circumvent the resistance. Several mechanisms that contribute to endocrine resistance have been identified. Metabolic rewiring is frequently observed in most cancer cells that exhibit resistance, and targeting glucose metabolism with well-established glycolytic inhibitors has shown to enhance the sensitivity to endocrine therapy in breast and prostate cancer models. The mutual interplay between glucose metabolism and androgen receptor, AR, ER signaling implies the combination approaches of endocrine therapy with metabolic modulators could be a standard of care to overcome resistance. Should be a standard of care to overcome resistance or even prevent resistance in the first place. Dietary interventions that modulate glucose metabolism have been demonstrated to be an interesting strategy for evading resistance to therapy. Well-designed clinical trials are urgently needed to elucidate the clinical utility of strategies mentioned above and develop metabolic drugs as routine standard of care in endocrine-resistant cancer patients in clinical setting. This was published in December of 2023. This is an urgent call that is recent for, I don't know if I disagree with that it needs to be drugs per se, the development of drugs. I think that for clinicians to either learn how to put people on ketogenic diets, to work with dietitians that know how to put people on ketogenic diets, that work with health coaches that need to put people on ketogenic diets, there needs to be an urgent training of physicians. There needs to be an urgent training of other clinicians in the field to help implement these dietary strategies first and foremost before you add the drugs, in my humble opinion. And it's not rocket science. It's really not. It's not that hard. It's not easy. I'm not saying it's easy for a patient who doesn't have any idea about nutrition to implement these things. For that, I do think they need guidance. But I also think that those patients, when they're motivated enough, they can figure it out on their own. And that is very apparent by people that I've worked with in the last 6 to 8 months. People who want to survive, they learn this stuff. They learn it fast, and they learn it better than doctors and other people. Going down further, targeting glycolysis to improve photodynamic therapy. So this paper is very long, and there's a lot more to share. In fact, I've got about another 10 slides. I think what I'll do is I'll take a breather here, and I'll finish this paper, and I will touch on the natural inhibitors and non-natural inhibitors, basically all the inhibitors that we've learned so far that I was able to find, at least, that have effect on the hexokinase enzyme. Okay, we'll talk about those as well as some of the drugs, but I think that we're already an hour in essentially, and I don't want it to be too overwhelmingly long for people. So therefore, let's take a breather and cut it off. And I've got a lot of comments that I need to get to anyway. So let me take a look at those. So to Glenn, who sent me an emoji that looks like this, welcome. I have uveal melanoma and I've lost my eye. I'm so sorry to hear that. Oncologists believe it has attacked my liver. No cure. I'm tackling it with ketovore diet, lots of holistic therapy. Any advice? Well, I can't give advice here. That would be a little unethical. But that being said, I haven't, this whole channel is built for you. Okay. This entire channel is to give you as much information and tools in your tool bag as possibly, to possibly help yourself. And then if you need additional help, find an integrative practitioner who has a clue about these things and start to work with them. If you don't have anybody locally, I can do educational consults, via Zoom, but it's really better, honestly, to have someone local, in my opinion, because they can order labs and they can help you and stuff like that. And then, just be a voracious reader as well. Start to learn this better than anybody else. And you're going to have much more success than just telling yourself that there's no cure and there's nothing they can do for you. I think that this is the time to really get serious if you're not already. It sounds like you already are if you're on a kind of a keto carnivore diet. I don't know if you're tracking. Maybe one thing I would say is track your glucose ketone index, track your blood glucose. If that means that you're using a continuous blood glucose monitor to do that for a period of time, I would highly recommend that. Because if you don't know how deep into ketosis you are, I don't think you're going to have good effects at implementing other parts of the therapy. I will leave you with that. Alan says, definitely sodium bicarb periodic daily intake is a critical element in reducing that lactic and succinic acid microenvironment to get. That's interesting you say that. Are you checking your urine pH yet? And if so, have you seen a difference in adding sodium bicarb? And then the next thing I'd ask you, as you see here, is that I don't know if I can't recommend, but I don't think I would personally be using Arm & Hammer. I think that those baking sodas in particular are known to have a decent amount of aluminum, and I don't want there to be any toxicity. So that's why I personally, again, I don't recommend these things, but these are things that we use in medicine all the time, is this sodium bicarbonate tablets. They're dirt cheap also. And then you get also a very specific amount of bicarb per tablet, usually 650 milligrams per tablet. The literature that I saw suggested that people need to go upwards of like 3 to 5 grams, which would be 3,000 to 5,000 milligrams per day to get the urine pH where you need to be. I think it's going to be on a case by case basis, obviously, which is by testing, just like the GKI is going to be critical for urine alkalization therapy. But I do think it is critical. I think by itself, it doesn't make sense. But I think as a way to enhance your ketogenic diet and metabolic therapy, I think it makes a ton of sense, which is why I had to get that kind of urgent video out. I think it's going to be a game changer. How do I intake this? How do I intake this? I'm not really sure what you mean by that. How do I intake this? I'm looking back at your previous comment. I may need some clarification for that one. Let's see here. Please list again the non-cancer cell. So please list again the non-cancer cells in our bodies that can absorb or take away or deprive glucose and glutamine from other cancers. Dirty little hands. Yeah. So to be honest with you, it's pretty simple. It's muscle. Okay. Muscle. So when you exercise, you're stealing glucose away from the cancer cells. When you are exercising with resistance training, probably more likely than cardio, you're going to take glutamine away. When you are exposed to red light therapy, that will enhance glucose uptake into cells and steal it away from cancer cells as well. When you are out in the sun, you're doing the same thing. When you are, or probably even more, to be honest with you. But when you get outside, you're doing the same thing. When you are exposed to cold therapy, that will steal glucose away from tumors, likely into brown fat. But when I went through the literature and I did that video presentation, it said that it was also muscle was actually a big part of it. So muscle, brown fat, that's where most of it is being upregulated in other parts of your body. That's why when I was looking in the last video at glucose transporters, and I was saying that, some of those substances were actually upregulating GLUT4, I was kind of okay with that, because that's actually what allows those substances to actually lower blood glucose when you're using them as glucose kind of sparing almost as like anti-diabetic or anti-glycemic medications, especially like Simularin and berberine, those kind of things. They upregulate GLUT4, and that allows the other tissues to steal it away. I just put a tablespoon of good old Arm & Hammer. Okay, I've addressed that. Pulse water fasting. Obviously, it's an N=1, Michael. Not everybody can do fasting. Not that it's difficult or that it is difficult for kind of everybody, although it does get easier. It's more like the weight and your kind of clinical status. I think it's going to be an N=1 and really kind of need to be talked about by your own personal doctor. But if you have the weight to lose, then it probably could be a good idea. What do you think of destruction of blood sugars via cavitation forces simultaneously building bone? Stuart McGill. You need the minerals from highly acidic stomach and into fixing bacteria. I would say that I don't know much of anything about that. I would say that, a lot of the ways that fibers and probiotics work to make you healthier is that it will improve glycemic control through a variety of mechanisms. In terms of cavitation forces, I'd have to look into that more. I don't know. I don't know the answer to that. Sorry. Doctor stated cancer cells do not use ketones. Dr. S, I'm guessing you mean C-free. Yeah, I know. But I know he says that, but what I want to give you is the devil's advocate that there are people out there who believe that some cancers can use ketones. But my point is that the word use has there. We actually have to pick apart the word use actually, because cancer cells can and do use fatty acids, right? But they don't use them for fuel. They don't use them to make ATP, right? They don't ferment fatty acids, and they don't have the mitochondrial capacity to actually burn them in the electron transport chain. So they're not using them. And when he's using the word use there, he's right. They're not using them for fuel, but they can use them to make other fatty acids, right? And to use them as a backbone, right? Which is kind of how statins work. Statins block the creation of new fatty acids, in particular cholesterol derivatives, through the blockade of the HMG-CoA reductase enzyme. And there's benefits to using statins in cancer therapy, probably again, not as a standalone, but as a enhancement. And it doesn't mean that you're blocking fatty acids so that the cancer cells can use them for metabolism. Well, energy metabolism, but those fatty acids can be used to make other. I mean, if you look at a cell, like what is the cell? It's surrounded by fat, right? All the organelles are surrounded by fat, right? And so my point is that cancer cells are using fatty acids highly to make new membranes, to make new whatever, and therefore fatty acids are being used by cancer cells. There's no doubt about that. It's just that they're not being used to make energy, right? And none of those processes to take one fatty acid to convert to a next, or one carbohydrate to convert to a next, or one protein and amino acid to convert it and build new proteins, they're going to require energy, right? So if you shut down energy, it isn't, to kind of use an analogy, if you don't have coal in your coal plant, to burn, to make energy, the electricity, it doesn't matter what kind of power lines you got, right, going away from the coal plant. It doesn't matter what kind of car you're driving. If the gasoline and the oxygen from the gas and from the engine are gone, it's not going to work. It doesn't matter what kind of carburetor you got. It doesn't matter what kind of exhaust you have. It doesn't matter what kind of wheels you got. It is not going to work, right? So I do think that the word use is kind of vague sometimes, and it's scary too. Let's be honest. When you see somebody or a paper suggesting that, prostate cancer can use fatty acids, it makes you think that, well, maybe Seyfried's wrong. Maybe Seyfried forgot to look at prostate cancer. Maybe Seyfried's just, out of it, doesn't know. I don't think that's really the case. I think that the word use kind of gets thrown around, and I think you got to really look at what do they mean when they say it, because it's not meaning energy metabolism. That I can say with near certainty. I don't know everything, so therefore, I'm willing to be wrong. It's just that every piece of evidence I've seen, I've not seen that cancer cells are using anything but glucose and glutamine for their metabolism of energy. Let's see here. Dr. Peevler, as you mentioned strongly, do not intake sodium bicarb within an hour of eating, nor an hour after eating. You need stomach acids to do their job and get neutralized by the carb. I agree with that fully. That's kind of my stance. I think that hopefully, what I would say is that I would want to take in sodium bicarb probably as infrequently as I could, right? To get my urine pH to where I want it to be. The more frequent throughout the day, the more likely you are to probably interfere with food, right? But also maybe the more frequent you are, the more likely you are to reach a steady state. So there's probably pros and cons, but definitely avoiding meals for sure. But doctor, you said earlier that permanent state of ketosis would not be good for human health. But if cancer cells can survive on metabolizing fatty acids, is there any escape in sight? If cancer cells can survive on metabolizing fatty acids. So I think maybe I'm not explaining it well. So cancer cells can't use fatty acids for fuel, okay? And fuel, I mean literally to make ATP, or the energy currency of life, okay? They can use fatty acids and amino acids and glucose and some other things to make other molecules, okay? That's also metabolism, right? It's just that it's not to make energy. It's to make other molecules. So there is a nuance there. What I'm saying is, if you shut down energy production through the restriction of glucose and glutamine, none of the other cellular mechanisms are going to work. They require ATP. So I hope that clarifies things. I would also say that I don't believe that a person should be on a ketogenic diet for the rest of their life. I don't think so. I don't even think it's necessary, if you want me to be very frank with you. Or at least I hope it's not necessary, because I think that would be not only difficult to maintain, but also maybe there would be detriments, later. My point is that there needs to be an end game, right? And the goal is to kill as many of the cancer cells and maim it as much as possible. I think the way I look at it is kind of the way that Gabor Sommelier looks at deuterium depletion therapy. So you use deuterium depletion therapy nonstop for a period until you get the response you're looking for, right? That could be 6 months, that could be 8 months, that could be 9 months, that could be a year, right? But then once you're in like a no evidence of disease or a stable, no progression situation, then, what he recommends, and this is not exact, quoting Gabor, like what I'm saying, but what I've heard him say and I've read about is that they recommend doing, a month or 2 or 3 of deuterium depletion therapy forever, to help keep yourself in a state of stability with your cancer or no evidence of disease. And I think the same could be potentially used for the ketogenic diet, where, this is more in line with one of my mentors, Dr. Jack Kruse. He talks about like obviously it depends on the latitude for his perspective. So I don't want to like blanket what he says, but he talks about seasonally eating, right? So in the winter, in the colder months, depending on your latitude, of course, doing more of a ketogenic diet. And then in the summer months, when you have long days and UV present, then doing a higher carbohydrate diet in the summer, right? So maybe something like that, where like half the year, depending on your latitude, of course, you're doing a ketogenic diet. And we're talking about like long after you're in remission, long after you're in a no evidence of disease situation. I think that's where I would like to get people to go to. And then of course, you're always looking at things that could potentially be causing or contributing to cancer and taking that out of your life, right? So obviously not just like going back to the same lifestyle that led to the cancer to begin with. I don't think permanent ketosis is ideal. Now, if I had glioblastoma or metastatic cancer that has a horrible, prognosis, we're talking like months, I think that may be something that I would be willing to just accept, that I'm probably going to be a ketogenic diet for the rest of my life. Of course, I wouldn't want that for anyone, but if that's what keeps them around a little bit longer on Earth and with their family and loved ones, I think that would be a price to pay. Again, is that ideal for human health? Probably not, but it helps keep their cancer at bay. That's very aggressive. And anyway, what's N equals 1? Yeah, that's a great question. Sorry. So in a study, the N is the number of participants in a study. So like a robust study may have like 10,000 participants. And the more participants you have, generally from a randomized controlled trial, the stronger the data is that being generally true, which makes sense, because when you have more people and it's generally seen as a benefit in the study, then it's going to be generally true. But we're not all the same, right? So instead of having an N equals 10,000, N equals 1 means like, this is what it is for me, and this is what it is for you. Everybody's different to some degree. Everybody responds differently, and everybody is in a different environment, right? So everybody is kind of their own N equals 1. I hope that makes sense. What is the diagnosis of HPV-associated left base of the tongue carcinoma with large ipsilateral left with a diagnosis? I don't know if that's like a continuation of the question of what is N equals 1. So I would say that there could be, let's say there's 10,000 people who have HPV-associated left base of the tongue carcinoma with ipsilateral lymph node involvement. Every person's environment is different. Every person has a different medical history. Every person has different genetic makeup and epigenetic environment. So therefore, everyone technically is an N equals 1. What I will say is that the N equals that the generalization that we can make is that all cancer cells rely on the Warburg effect and aerobic glycolysis and massive amounts of glucose. All cancer requires glutamine. So, and all cancer cells will upregulate their glutaminolysis and glucose, not only for energy, but also to make glutathione to help protect themselves against reactive oxygen species. So therefore, the non N equals 1 is metabolic therapy as laid out by Seafried. Where N equals 1 gets in is looking at your exact environment, looking at your exact makeup, your age, where you're living, what your latitude is, what your job is, what is your sleeping, what is your bedroom look like, and what I mean is like, how is your sleep environment and how is it restoring your health versus destroying your health? That's the N equals 1. And that's where, in my opinion, a doctor really feeds into. A doctor's job, in my opinion, other than make a diagnosis, is to help figure out what makes you special and how did you, in particular, develop this disease, right? And nobody is going to be able to tell you exactly, no one's God by all means, but there are some doctors who are more awake, I'd say, to looking at the environment and what can potentially drive damage. And part of our job, I believe, is to highlight those things and then help you get on to a metabolic therapy, help you mitigate your environment, or put you in an environment that's going to be leading you to maximum health. Fasting and ketogenic diet for the push part of, but need the glutamine inhibitors for this condition. Every cancer needs glutamine inhibition. Every cancer. So no one is unique there if you have cancer. Cryotherapy, dry, wet, 180 degrees, 3 minutes. I can't speak for it. What I could tell you is my mentor, Dr. Cruz, doesn't believe that cryotherapy is going to have near as an effect as cold water immersion is going to have. And it's not going to be cold water immersion as highlighted by like influencers and like health anti-aging people, where you're doing like freezing temperatures for like 30 seconds or like 3 minutes or something like that. We're talking like a more reasonable temperature over longer periods of time as a way to upregulate brown fat maximally and steal the maximum amount of glucose out of the system. It'd be like exercising for 30 seconds, right? Or 3 minutes or something like that once a day, and expecting that to be the same as, doing a real resistance training. I think it's just, I don't think it's going to be the same. Sauna. And then cold shower for brown fat for 20 minutes. Yeah, I mean, I don't know, to be honest with you, the answer to these questions. I think these are all like good strategies as a way to potentially enhance, your body's function. I'll be very frank with you. Although, Finnish saunas at that 180 degrees Fahrenheit are what's talked about a lot by, most notably like Rhonda Patrick and like those kind of folks who are like kind of like sauna experts. If you look at the literature for sauna therapy and cancer, you're going to come up with like zero results. All the literature is looking at cardiovascular therapies or cardiovascular diseases. Do I believe that there's benefit to it? Absolutely. I just don't have any data to back that up. What I would say is there is one study that I found on infrared saunas and infrared light therapy, far infrared light therapy, excuse me. And it had profound effects on, I think it was a murine or a mouse model with, I believe it was hepatocellular carcinoma. And it had profound effects on shrinking the tumor. It didn't really get into, exactly like how many, temperature and kind of like strategies to implement for humans. But I do think that there's benefit to sauna. It's just that there's just not a lot of data. So therefore I really can't comment, unfortunately, a lot. Temp. Yeah, those are all questions we don't know yet, unfortunately. How soon is the sequence after hot and cold therapy and vice versa? That I don't think I also can answer the question to that. I don't think really anybody knows. I think protocols are being built. What I would say is that we definitely seems like from what I like general principles, it seems you want to do cold therapy. If you're going to do cold therapy in a day, you do exercise. You definitely want to do it before exercise to not blunt the benefits of exercise, and especially resistance training in particular. I'm not sure about cardio, but definitely like weight training, resistance training. It seems to blunt the effect if you do it after resistance training. So I wouldn't do that. And then sauna therapy seems to be better after training. And I've seen it to where it has an effect right after your training session or later in the day on the training session. And so therefore, like, you know, I don't know the answer completely. What I'll say is that I have a sauna in my garage right next to my garage gym, right? And so for me, the ultimate convenience would be to do it right after my workout. I personally don't do it. I've done it right after my workout. I feel better if I do it, you know, several hours after my workout. Am I limiting benefits to it? I don't know. I think that there needs to be a lot to be discovered with like timing of these things. And I don't propose to be an expert on that. So I think that I'll have to either look at other resources or whatnot. Sorry, for cold water immersion, what temperature is ideal? So I would refer you to Dr. Jack Cruz's cold thermogenesis protocol. It's free on the internet. You search cold thermogenesis protocol, Dr. Jack Cruz, and he'll have like the way that you get yourself set up for it. I think that it's kind of a little bit antiquated at this point because I think he published that protocol back in like 2012, 2013. Not that it's not good. It's just that now we didn't have cold plunges that we do now, right? Like right now we can have like literally like a little switch or a lever that I can put, okay, I want to put it 70. I want to put it 65. I want to put it 60. I want to put it 32, right? Like I can have exact precision with the temperature, right? So it doesn't require the same strategy that we did, you know, 15 years ago when we didn't have those tools. So what I would say is that likely the target temperature is going to be somewhere between like 50 and 60 degrees Fahrenheit, probably, but that being the goal temperature. But it's not for like 3 minutes like you do when you're like diving into ice. It's going to be more like longer sessions where it may be like 20 to 30-minute sessions. And, you know, maybe you could even do it more than once per day. But I think that, yeah, I probably wouldn't go more like colder than like 55 degrees Fahrenheit. I don't think it's necessary. And you can stay in there for longer. Now, if you want other benefits, like let's say aside from cancer, then there could be other benefits of being in like that really cold water for like a shorter period of time for like, you know, endorphins and like, you know, boosting dopamine and serotonin and norepinephrine and other things that can help you feel good. But in terms of like for a practical, you know, things we can do with cold, I think probably longer periods of time is going to be better. And probably not as cold, but I think that still needs to be elucidated. Thank you for the super chat, Michael. I appreciate it. And, you know, we've got an hour and 12 minutes. I'm glad I stopped right here at targeting glycolysis to improve photodynamic therapy because, wow, we would have been like an hour and a half or longer if I would have hit that. So let's just plan on the next video being to talk about, you know, glycolysis, as well as some of the natural and drug inhibitors of hexokinase 2. And then probably the next video after that, I'll specifically hit a very important hexokinase inhibitor called 2DG or 2-deoxyglucose, something that I have toyed around with a little bit. And I think that there's a ton of potential. It's just that I think there's also downsides to it because it's such a strong inhibitor of hexokinase. Even if you're in therapeutic ketosis, people tend to feel hypoglycemic. So anyway, cool. My oncologist is pushing me towards radiation chemo. Yeah, I know. Look, they do that because that's what they know. You know, I don't think there's any malfeasance from that. That's what they know and that's what they understand. And they probably would feel anxious if you didn't do that. That being said, there's obviously more to the story than just the conventional therapies. That's why this whole entire channel exists. And I'm sure there's other channels that have other information about other alternative therapies. And it is you, the patient, that is to decide what is right for me. And it is not anybody's job, no doctor. You know, what I learned when I was in medical school early on, like my first year of medical school, they taught us autonomy first. Our job is to present information that what we understand is to be true or to be important, and present that in a way that is, you know, hopefully not not pushing, but persuasive enough to help a patient, maybe accept that treatment as being a good thing for them. You know, not shaming or scare tacticking or pushing or telling him I'm going to fire you from the practice if you don't do what I want you to do. That is not really the art of medicine. That could be considered a dark art of medicine, maybe, but the art of medicine is really saying, look, I have a unique knowledge here, and I believe that this is to be the best thing for you. And that's how it should be, right? But ultimately, the patient, no matter what the treatment is, whether it be Eliquis for DVT or whether it be, sodium bicarbonate therapy for alkalinization of the urine from an alternative perspective or ketogenic diet, like ultimately the patient has to make the decision, and we shouldn't be pushing anybody into doing anything at all. But I do get that some people get pushed and shamed and all those things, and that's horrible, and that shouldn't be. And I'm sorry for that. 90%, 90 fat, 10 protein eating, processed sugar, but not true fasting, is it? It is not water or dry fasting. No, it's not the same. Not completely the same. I would prefer to buy an advanced longevity program if you're able to publish or sell one to people who are not ill. Zigzag, I actually did a kind of a trial of I forgot what I even called it. I called it like enhancing mitochondria or something like that. And it was a I want to say I did like 3 hours total. It could have been maybe slightly more than that. It wasn't quite as long as my cancer course that I did. But it was basically enhancing mitochondria, obviously removing harm and enhancing the mitochondria. I did something like that. And it just didn't have a lot of, there was I think it was like 4 or 5 people, and we had a great time, don't get me wrong. And I love presenting the information. It's just that this channel seems to attract people who actually have illness, and I'm okay being there for them for that. Although I do have a passion for preventive medicine. So I think that if I can get enough people who are interested in it, I'd be happy to present that information again. It's just that there's just not a huge amount of demand compared to people who want to know about metabolic therapy for cancer. A comment about Maurice Israel, alpha lipoic acid plus hydroxystrate plus methylene blue. Interesting. There's a lot of papers out there that actually combine alpha lipoic acid and hydroxycitrate for metabolic therapy, and I'll have a whole video or a couple videos on them. Thanks, mate, from down under. I think it's so cool that I can talk to people from Australia via live video. It's pretty cool. All right, so I think with this, we're going to shut it down. Thank you for being part of the live stream. I hope this was helpful for you. We're going to hit every important piece of glycolysis going forward. We still have a lot to cover for the hexokinase enzyme in particular, but we will cover all the pertinent enzymes that have, strong rate-limiting effects, and we're going to cover them all in a vacuum, but then we're going to zoom out and see how we could use them potentially in combination for maximum kind of press pulse therapy on the Achilles heels of cancer. So until next time, I hope you like this. Please like it. If you have people that could utilize this information, please share it with them. And if you haven't subscribed, it is free. Please go ahead and subscribe, and we will welcome you to the Mitochondriac Army. Until next time.